Thanks, Taryn. As Taryn said, I want to thank everyone for coming out today. Really appreciate it, and your excitement to participate in research, which is what I focus on. And so my role at the Barrow, so I was recruited out here 14 years ago to sort of build an ALS research center here in Arizona. So there wasn't much going on in ALS research back then in the state of Arizona. And so with working with Dr. Lada, created a new clinic and have continued sort of to grow the program. So we're clearly one of the top centers now in the country in ALS and perform a lot of research activities and run a lot of the clinical trials that are ongoing throughout the country and around the world. So incredibly excited about that. When I start talks, I always have to put up disclosures. None of them are relevant for this presentation. But really, you're here today because of Target ALS, their involvement, and their interest in developing these what are called community-based pop-up clinics. And so what you did today was sort of participate in a research effort around a pop-up clinic where you could answer questions, be consented, provide a blood draw. And that is a fantastic inroad into research if you've never participated in research before. And it's really critical that we get these samples and clinical information for our research studies. There's a great need for continued data from patients and controls, like all your spouses that were participating. They're really important because that's how we compare ALS to somebody without ALS. What's different? How do we gain more insights into the disease? And so I'm going to talk a lot today around some of the work obviously ongoing here locally and some of the studies that I help run at a national and global level. And first off, I just wanted to mention, I'm sure no one is reading and scouring the scientific and medical journals, but last week there were publications around the first 4 Healey platform trials. Everyone know what that is? The platform trials? Yeah, so they're phase 2 trials testing compounds for 6 months. And the results, well, have been known for a little bit of time. This is really the peer-reviewed publications coming out that everyone can really look at. And so from those first 4, 2 were completely negative. One trial was halted due to futility. That is, they looked at it halfway through. It was not going to hit any of their endpoints. It wasn't working, so we could stop it for futility. That's actually really good when we can do that, because then you can move on to another trial. Plus, it saves a lot of money and effort from all the folks involved. And the second here on the bottom, really the results after 6 months were just showing it wasn't hitting any of the outcome measures, really wasn't changing progression at all. So another negative, completely negative trial. Then there were 2 that especially the sponsors were really interested about, all right, this kind of warrants further investigation. And the first one was a compound from a company called Clean Therapeutics there on the left. And from this 6-month trial, the results, it wasn't changing the clinical parameters of disease, but when they looked at some of the biomarkers, including neurofilament, which you might have heard about, what they noticed was that people that were receiving the drug didn't see an increased rise of it over time that we often see in patients. And so they were very interested in that. So this biomarker is released from neurons that are injured or dying. It builds up in your cerebral spinal fluid, and then it goes out into the blood. And so we can measure it in either of these biofluids. I've been involved for years in studying neurofilament and really early proponent of really pushing this in the clinical trials arena. And then the second drug, again, in the 6 months, wasn't showing changes in progression, wasn't hitting the typical clinical outcome measures that we use in clinical trials. But in this case, they were using speech as a measure. And what they noted was that by speech, there was a slowing of progression that maybe you couldn't see yet clinically, but they were very intrigued by that. And so both of these companies are interested in further pursuing those. So Clean is the one on the left, the CMU AU8, and Perlinea. Perlinea is the company involved in the second. So that was really exciting for the community, and we can really now dig deeper into those results. And there's more to come on continued HEALY platform trials that are ongoing. And now looking sort of more locally at the Barrow, we have a lot ongoing in sort of both basic, translational areas of neuroscience, and a lot in clinical trials and clinical research. So we use a lot of patient-derived models and samples. So blood CSF, the blood that you provided today, might be used in some of our studies. We also use patient-derived stem cells. So we can develop those from blood. We can develop those from a skin biopsy. But they're also very useful because now you can, and I'll show you in a few slides, you can create sort of a model of a person's disease in a dish. You can test drugs then on that. And then in the clinical trials, again, we're definitely a leader in this country in clinical trials. And so it's incredibly exciting to be heavily involved in the drug development process and the trials that are coming and becoming available to ALS patients. And a lot in biomarkers as well. So again, that's what I've worked most of my career on, and really developing different biomarkers and types of biomarkers that can be useful in monitoring disease progression, helping to tell whether or not a drug is effective. So just mentioning a few slides, these I got from Rita Sattler. She's one of the scientists at the Barrow, who's exploring, again, ALS using human model systems. And what are these model systems? Some of them are, yes, question. Can you just explain what translational means in this context? Absolutely, great question. Yeah, if you got questions, just yell them out or raise your hand. Yeah, so translational in this concept means discovering things like in the research setting at the lab, and then trying to translate them, move them into the clinic. So really focused on discoveries, and then taking those discoveries to develop new treatments that we can then provide to patients. So that is sort of the translational aspect. It's incredibly hard, it's incredibly expensive, but that's the goal. And then often when we get these into early stage studies, then we really look for industry sponsors. We really look for industry partners that want to then take this to the next level, because that's even more costly. And so that's where industry comes into play. So what types of human model systems do we use? And again, on the top there is using tissue. And so through Target ALS, I'm a co-director of a national tissue banking program where people volunteer, and we collect tissues and then provide those as a research tool to scientists and investigators throughout the world. So that's one area. Another area is, again, using biofluids from patients, using imaging. So we do a lot of imaging studies on ALS patients, as well as we're learning how to use imaging as a tool to monitor progression of the disease. And then the last one on the bottom there is, again, looking at sort of patient-derived cells in a dish. And so in Rita's lab, they do a lot of this work, and it's really taking patient-derived cells from individuals, sporadic patients, those with specific mutations that are causative of the disease, and then growing the cells that are normally within our central nervous system, within our brain and spinal cord, and then asking questions. What's changed? What's going on that's different between these same types of cells coming from a healthy individual, for instance? And so Rita's lab is heavily involved in this, and this is just another slide from her lab showing the main three types of cells in the central nervous system are neurons up there on the top, astrocytes in the middle, another cell called microglia, which is on the bottom, and just some of the results. Again, these are actual cells that are growing in a dish in her lab, all developed and induced by patient-derived stem cells. Some of the questions that she's really looking at is, again, very basic. What's going on and how neurons communicate with one another? And they communicate through this specialized area called a synapse. And the other cell types involved, the astrocytes, the microglia, they really help out in pruning and making sure that synapse functions properly. And so too many or too little of these cell types and the synapse doesn't fire right. The cells don't communicate. When cells don't communicate well together, often they'll die or retract. And so you'll lose those communications. And so Rita's lab is really interested and focused in how these types of communications are not properly working in patient-derived cells from patients with ALS. And she can see that even with a sporadic cells generated from sporadic cell lines, there's a difference between the synaptic connections from healthy individuals that provide stem cells. And so it's really looking at the basic biology, because once we uncover that and know that better, we know what to target with drugs, and we can try to reverse that. So that's one area. Another one, another investigator named Dr. Bakar, shown here, she's interested in another area, which is the barriers from the periphery, from say the bloodstream, into the central nervous system, into the brain and the spinal cord. She's been focusing a lot of her efforts on one of the barriers called the choroid plexus, which there's no test at the end of this, so you don't have to know much about it. But on the image, if I can get the little pointer to work, maybe I could highlight it. There we go. So on this image here, the choroid plexus lies in these areas here highlighted in red, and it forms a barrier again, now between the bloodstream here in a very tight layer of cells into the cerebral spinal fluid. These cells secrete a lot of growth factors and other molecules that are critical for how neurons survive throughout the brain and spinal cord. And so Dr. Bakar has been very interested in look at, are there differences in somebody with ALS from somebody without ALS at the choroid plexus? And sure enough, she found a lot of alterations of the choroid plexus. And so it's been identified now as a new therapeutic target. No one's ever looked at this before. And so now we're thinking, how can we target this specific barrier to work better? And so hopefully slow down and impede the progression of disease. But there's lots of challenges in ALS research. As you're well aware, it's a rare disease, so the incidence is low. It's very complex. We know over 40 genes that are involved, and that number continues to grow. Some of these are causative. Mutations cause ALS. Some of them are risk factors. Changes actually just increases the likelihood that you might develop ALS sometime in your lifetime. So we're learning more and more from the genetics of the disease. But unfortunately, most of the people that participate in all our research studies, in our clinical trials, most of them are about 95% Caucasian, Northern European descent. So we have to do a lot better in bringing in more diverse populations into the research setting and into our clinical trials. And so that lack of ethnic diversity, I think, is skewing what we're studying and what we're trying to find cures for. So there's challenges depending on the community with just lack of trust with the medical profession, and for good reasons in the past. Sometimes even the primary care physicians, especially out in rural settings, never really refer anyone to a neurologist or an ALS specialist throughout the whole continuum of their disease. It's a disservice to the patient and the family, but again, it's what they're practicing in rural settings. Oftentimes, we come up with individuals can't travel long distances. We're in a big state. And so if you're living in a corner of the state, it might be really hard to come down to the Barrow, and gets harder and harder during the course of disease. And so that's another barrier. And then sometimes patients will come once and just tune out. They don't want to participate. They don't want to know anything more and just prefer to stay home. Another big challenge is the lack of, as I mentioned, longitudinal samples for the disease. So how can we track how the disease changes over time if we don't have clinical data and sort of blood and CSF of patients during the course of disease? So today you gave one blood sample, and I thank you so much for that. You're now one of our research participants through Target ALS. We have other programs that I'll mention in a few minutes where you can enroll and continue to provide information and samples. So again, we're possibly missing really important genetic information on what causes the disease by really focusing on Northern European descent individuals. And we know this because if you talk to most people in the field, they'll say that this repeat expansion in this gene called C9orf72 is the most common genetic form of ALS, and also frontotemporal dementia. It can go either way. That was an incredible discovery about 8 years ago. And, it's been shown over and over again, in the United States, at least from everything that we can see, and definitely in Europe. When we moved that over to Asia, our Asian colleagues said, I have no idea what you're talking about. We never see this. That mutation doesn't exist in the Asian population. What's the most common genetic form in certain ethnic populations? We have no idea. So we're missing out on a lot by only studying a very finite group of individuals. So we got to do better. And so luckily for us here locally, we've been the recipients of funds to help spearhead a lot of these efforts. So a lot of the largest ALS clinical research studies ongoing in this country are all run out of the Barrow. Funding from Target ALS, and so you're familiar with Target ALS today. Funding from the National Institutes of Health, NIH. Funding from the Chan Zuckerberg Initiative, Zuckerberg, Facebook. Him and his wife formed their own funding agency especially for rare diseases and for diseases in underserved populations. And with Chan Zuckerberg, with the help of Target ALS, I wrote a proposal to say, we're doing a horrible job at reaching out to communities. Here in Arizona, we have a large Hispanic and Native American population. I want to try to build a program to reach out to these communities. And so Judy, who you might have met back at the Target table, is somebody that I hired directly from some of the funds from Chan Zuckerberg Initiative. So with Target ALS, it's running this global natural history study and longitudinal biofluid study. So on the map on the right, you can see little dots, bluish dots of sites that are active. So you see a bunch in the country, not surprising, but now we're starting to go south into South America. And so we have sites there. We have one site in Israel, one site in South Korea, and then bringing on sites in Malaysia and India, 2 sites in India that will be coming on board. Incredibly exciting because this is really going to push the boundaries of the types of individuals we bring into this study. And again, by far, it's the biggest global effort to try to recruit patients into this longitudinal research study. So we already have about 200 patients that are enrolled in this study. We have lots of biofluid samples, over 17,000 vials that are sitting in freezers down the street or up the street, depending on, it's about 2 miles that way, the Barrow. And within this, about a year ago, Target ALS created a web portal where all the data, all the information around the samples and the clinical data it resides. And so people around the world can then apply to have access to everything. And so we're already giving out lots of samples to investigators and the companies around the world for their research studies. I mentioned, the sites in America. Here's just sort of the listing of the 14 active ones. And again, lots of different sites around the United States, but again, reaching out in a growing global effort. So there is a quiz on this later, so you have to know the 14. So in this study, what are we really doing? We're collecting clinical information in a longitudinal manner when you come into the clinics, one of the clinics. We're collecting biofluids, blood, and cerebrospinal fluid and urine in the clinic. All those materials go to the Barrow regardless of where the site is, and so we hold it as a central repository and then ship it out to anyone that wants it through the Target ALS portal. We're also doing additional measures, sort of at-home measures. And so we've done speech measures with 2 different companies. So the great thing is you start to get a larger and larger project ongoing. There's a lot of companies out there that want to use speech as an outcome measure, as a measure to, or as a way to look into and gain insights into disease progression. And so often any one study, maybe you can use one of these companies to be involved. But when you start to build a large program, so we're trying to have 800 ALS patients and 200 controls throughout the world, we could actually have multiple companies in different types of technologies that are all just built in. And so we can collect information from 200 with this digital health measure or speech outcome measure, 200 with this, and so forth, all collectively built under one umbrella and one infrastructure. Incredibly valuable and incredibly powerful for downstream analytics. And again, it's really getting all this material and information out to investigators as quick as possible. And that's the key thing. I mentioned the Chan Zuckerberg Initiative. And again, this was an outreach program. Something else that has been very, it really hasn't been done in ALS, surprisingly, is outreach. It hasn't been done in an organized way. People try to do it, and people want to do that at their site, but a very local, sort of focused manner in how to do outreach. And so through some funding from CZI, I hired Judy. And so she was set out and tasked with a lot of different goals and really outreach to the Hispanic and Native American populations. So she spent the first year really just connecting with community leaders. I mean, the only way you're going to build trust is to meet with leaders. It might be at a community center, it might be at a church, it might be wherever, but you just have to take some time. And actually, that was the key thing, I think, when I was talking to the Chan Zuckerberg Initiative folks. It was, yes, I want to do this. No, we're not going to have great results in a year. It's going to take time. And so you have to trust us that we can do this, but it's going to build over time. And so it's a lot of community education. Taking our documents that, I only write in English. I took Latin in high school. It's not very helpful anymore. But you have to translate all the documents into whatever language you need. All the consent forms, all the information has to be in multiple languages. And so it sounds easy, but then, we function in the medical arena, so it all has to be approved by a group called the IRB. And so they have to approve all these forms. You can only do it with certain companies. So it's a time-intensive process. So again, for Judy, it was trying to increase diversity within our ALS research programs and clinical trials. And she's also working with various other organizations that I'm sure all of you interact with on potentially a daily basis, on other sort of patient-facing organizations, I Am ALS, Everything ALS, et cetera. And so really reaching out to them to get word out. How can we reach more people quickly? So the goal, again, removing barriers to participating in research. And so I've already mentioned a few. Another one that I just want to mention, and I don't have my reading glasses on, but I think it's here. Another important measure or fact is that, again, travel's often hard. Target ALS really stepped up to the plate, and they said, we'll fund travel for people to come to your clinic. So you might be 4 hours away in Arizona from here in Phoenix. They'll fund the travel of the patient and their caregiver. We'll fund them to even stay overnight if they need to stay overnight, fund all their food, their lodging, and travel costs. It's amazing. But again, it's just breaking down traditional barriers to being involved in research. So now I'm going to mention another big study. So this is called All ALS, the Access for All in ALS. Has anyone heard of that yet? No. Okay. Yes and no. It's about right. It's fairly new still. So this was funded from the National Institutes of Health, and it got kicked off a little over a year ago now. And it was a big effort and a lot of great applications to NIH around this from people around the country. So at the end of the day, we had 2 awards. One, me here at Barrow as the West Coordinating Center for this effort, and a friend of mine, James Berry, who's at Massachusetts General Hospital, to coordinate the East Coordinating Center. So here we have 35 clinical sites across the country. This is US only. So again, something that's different from Target. Target's going global. All ALS focuses just in the US because it's NIH money, it's taxpayer money, it's our money. So it's only in the US. But listed here are all the sites that are currently involved. And in all ALS, we have two protocols that are ongoing. One is called Assess ALS, which really is symptomatic and controls. So again, symptomatic ALS patients and healthy controls. Very similar. We're actually working with Target ALS hand in hand, which isn't surprising because I help run that. So it was really creating the efforts whereby these two different large programs are collecting similar, identical information, collecting it the same way. So at the end of the day, investigators around the world can take data from both sources and use it. And so it's the best use of materials and the best way to do research. So again, it's collecting longitudinal data and biofluids. For all ALS, CSF is optional. So in all ALS, we're trying to include, NIH has told us, nearly every ALS patient in the country. That'll be hard. Some people just don't want to do research. But in order to do that, then CSF is optional. If you don't want to do it, that's fine. We'll just collect your blood. Another difference is we have fully remote. So if we're going to collect samples and information from everyone across the country, if you live in North Dakota, where do you go? You're not coming here. It's too far away. So we have the capacity now to do it fully remote. And so we collect only blood, again, from the remote participants. So we have national phlebotomists around the country that'll just go to your house on a quarterly basis. And so that's how we're running the fully remote portion. So again, that's different from Target ALS as well. And then also a little bit different is we have another protocol, a second study called Prevent All ALS. And here it's looking for genetically at-risk individuals. So again, looking at the genetics of the disease and now looking at family members of those that harbor genetic mutations. They can either opt in to know their genetics or completely stay blinded to whether or not they are a carrier of the gene. And it's following them longitudinally. So now we're actually taking people that are healthy. They're perfectly fine, but yet they have a genetic alteration that might cause ALS down the road. And so the big questions for this study is how and when does that happen and why? Is it an environmental factor that triggers something? Is it an automobile accident or a surgery that triggers something that now results into ALS? And so it's really looking for how this, what triggers the disease? How does one convert from being asymptomatic to now being symptomatic? So this study, now we have a new website. We have about 250 people involved in it so far. And you can go online and even fill out a form and say, yeah, I'm interested in participating. And so we have patient navigators that will reach out to you and coordinate all activities from there. So those are some of the really big clinical research studies that are ongoing that, wonderfully I help lead. It's a tremendously fun opportunity. I mean, I can't express how much I enjoy doing this. It's really hopefully making a difference in how to accelerate research, how to accelerate the identification of more effective treatments for our patients. And at the Barrow, again, we're sort of clinical trial leaderships as well in the last couple of years, probably less than 2 years now. We run or participate in 12 ALS clinical trials, 6 expanded access programs. So if you know what that is, so a number of companies will continue to offer the drug in an expanded access program, trying to bring in people that aren't otherwise eligible for clinical trials and provide them opportunities to be in a clinical trial setting. And then also 10 other ALS biomarker and natural history studies. So some smaller ones, local ones, and including the couple that I mentioned today. The 2 today are the biggest that are out there. And then at the Barrow, our team is big and growing. So sort of on the left, these are sort of the laboratory investigators that are involved. And then they have their team, either having fellows that are within the lab, having students, PhD students, MD, PhD students, lots of undergraduates. We even have summer students. We have high school students that come in and help do some research. Then a lot of research technicians across the labs. Within the clinic, a couple of the clinical PIs, and we actually, in July 1, have a third who's coming on board at the Barrow. At the Barrow, the CRO, so Contract Research Organization, so Dr. Scheffner, who stepped down from seeing patients, he's now focused just in this effort, which is a monitoring effort. So he runs a monitoring service where he has a large group of people, I think it's over 20 now, that go from site to site or across the country and around the world during clinical trials and monitor them. Are they collecting what they should be doing? Is all the data that they're collecting accurate such that we can interpret it down the stream? And so this is a large endeavor, but it's the only one of its kind in ALS. And so the leaders of that, the 3 leaders of that are listed here. We have a biorepository team. I mentioned we have a central repository for the Target ALS program. So we have people that are involved in just receiving and shipping and making sure that all the freezers stay frozen. Big team that's involved in that because it's a large process. And then on the clinical side, again, a large number of program and project managers, the clinical coordinators that many of you, I'm sure, have been in contact with and communicate with. We now have 2 patient navigators, so we're excited about that, and now ongoing outreach programs at the Barrow. So really excited, and really, again, just a special thanks to all the patients and families that really step up, contribute, want to participate in research and clinical trials. We couldn't do it without you, and so I just want to say thank you. PhD students at what university? They come from, currently they come from ASU. So we have a joint PhD program with ASU. We have a joint MD PhD program with University of Arizona Phoenix, with their college. Creighton University, Creighton now has a medical center, and they want to start an MD PhD program, so I'm sure that's up and coming. And then ASU is going to be starting a medical school in the next year or so there as well, and so we're already talking to them. So that is all I had to say. Any other questions? Yes?
So when you go to clinicaltrials.gov, the ALS FRS Revised
has
such a central role as determining the evaluator of whether the approach was successful or not. Gosh, I wonder if you have a little bit of thoughts about that. Is it really that effective? Is it perhaps part of the problem why so many drugs don't seem to make it past?
Yeah, the ALS-FRS barrier, because that's usually the primary outcome measure in all clinical trials.
Yeah, it's kind of a complex subject, but maybe you can just make a couple of comments about that as the giant hurdle that is determining whether drugs become available therapies.
Yeah, I mean, it's a fantastic question. So can there be challenges around it? Yes. There's, even FluoroFax, you can only go so low, and in certain parameters. So you're all well aware of the questions and everything like that. And then they get segmented into certain areas, and we look for changes over time. So the challenging thing is, yes, there are, I wouldn't say issues, but it's the interpretation of that. It's not exactly, if you take 100 people and look at all their scores, it looks like a nice linear drop, but on an individual to individual basis, not so much. Now, there's been a lot of efforts over the last couple of years. We had initial challenges in global clinical trials because what we learned was that the Europeans weren't exactly asking the questions exactly like we did. And so we quickly ran into problems around that. Dr. Scheffner and some colleagues in Europe then created a harmonized went back and re-examined everything and created a harmonized version, so now we do. So, but that's only the last year and a half or so. So there are more issues there. The FDA wants more outcome measures. With all ALS, they want more digital outcome measures. So we've been charged with trying to help in that process. And interestingly, so we have continued talks with the FDA around this issue. And specifically for the ALS FRS, they really wanted to know what is the minimal difference that a patient says is significant. Surprisingly, we don't have that answer. We know it changes, and we usually have, if it's less than this number of, this point whatever 5 per month, that's a certain rate. But to a patient, what's truly significant? Because the FDA approves drugs, obviously based on safety, no toxicity, et cetera, but they're most interested in what is beneficial to a person living with that disease. And so as part of all ALS, we actually built in with the FDA a sub-study to figure that out. And so we've collected data on, I think, the first 100. And so we have to build up a little bit more. But they were very interested in using these natural history studies that I've just been mentioning as an avenue for the regulators to get more information and guide their future regulatory decisions based on this knowledge. And so I think as we move forward, we'll be able to provide the FDA with some really important insight as to what is that minimum difference, such that when they see the next clinical trial results, they say, well, this is what patients deem significant. If it's not meeting that threshold, then the answer is no. And so it's been a lot of fun in that regards too, working with the regulators and seeing how their mind works and what's most interesting to them. Sometimes it's different than what's most interesting to me, but it's they're the decision makers on approval or not for a drug. And so I think these discussions are really beneficial to the field at large, because as we work together hand in hand, they get to understand our disease a little better. We understand their thought processes better, and that's to the betterment, I think, of drug development for the disease. Yes?
There's been some recent research, not in ALS, but showing that humans can learn how to control their arms and limbs with their brains without when the nerve is being cut.
Yes.
Are they looking at that at all with respect to ALS?
There's been.
Muscles are normally okay in an ALS patient?
Well, you get muscle degeneration as well, because when the muscle loses connections to the motor neurons in your spinal cord, they degenerate, because as I mentioned, that synapse, that connectivity, that's important to keep both cells healthy. And so when one cell dies, for instance, then the other cell's looking for that signal and it's not getting it. It starts to retract and regenerate and degenerate because it's not receiving a signal from the next cell. So in ALS patients, there is muscle degeneration that's ongoing. But to your point, depending on the individual, depending on the course of the disease, obviously there's lots of muscles that are healthy, that are working just fine. But can you regenerate or repopulate motor neurons to the muscle? We're not there yet, but you can still do stimulation, and that's called brain-computer interface. It's been done in ALS patients in the past, and it's been done recently in ALS patients. And there are a lot of interest and a lot of efforts ongoing at trying to develop more and more brain-computer interface modes for different disease populations, and ALS is one of them. Yes?
The all ALS research program you have, that you stated they have genetic testing for family members.
Yes.
Patients that have ALS, so do my sons have to have medical insurance to participate? Or?
Nope, all free. So the program pays for it all. We pay for the genetic counselors, any type of services there.
So they just call and say we want to set an appointment?
Yes. Yeah, it can work If you're working with a clinical coordinator now, it'd just be working through that coordinator to schedule those. Oh, you could talk to him too, yeah. And then he'll direct you to the right coordinator for the program to set up those appointments. Yes.
Question, just yes, I think I heard you mention if family members do want to participate in the longitudinal part of the study and do the genetics and stuff for the sake of research alone and not for the sake of personal knowledge, is that an option?
Yes, absolutely. Yes.
If you don't have a genetic factor, is there any point in your offspring being tested?
Unlikely. So yeah, if there's no family history of especially ALS or frontotemporal dementia, even we look at any other neurodegenerative diseases as well. But if there isn't sort of that family history, and it really needs to be going down like through an arm of the family, you know what I mean? If the mother to the grandma, etc. If that's not present, then no. Yes.
So the catalog, I think you said, is about 40 for mutations
and
implicated genes.
Yes.
So you think that that's just getting started? Like, you can expect 4,000 over the next 25 years.
No, I don't think it'll be 4,000.
You think that actually it's quite mature, and you think you have the pulse of the right thing? It's just the interactions.
I think we have. I mean, it's still, if you added up all that 40, what percentage of the genetic forms do we know? We're still missing almost 30% or so. So we're missing genes. We definitely are. Many of them might be risk factors. It'd be hard to find. So that the number will grow. I don't think it'll get up to 4,000. But in other directions it's going to grow is that, we know that sort of 70%, 60 to 70% for Northern European Caucasian descent individuals. As we start to incorporate more and more people of mixed ethnic diversity, different populations, that number is going to change. I mean, so we're going to have genes that will be involved in specific ethnic populations. And we know almost nothing about that right now. Now, I still don't think it's going to get to 4,000, but it'll definitely grow. At least now we have most of the technologies we need to help do that and do that rapidly. Yes?
Well, I noticed in the questionnaire that we did today, the environmental one, they didn't ask anything about ethnicity.
About what?
Ethnicity.
Oh, well, because that was an environmental factor question. And I'm glad you didn't quiz me on everything that's in that questionnaire as well. It's really hard. I mean, I don't know if anyone has done the CDC registry, because they have all these modules and they're fantastic. It's just, a never-ending list of modules. So it matters how in-depth you want to go at sort of environmental factors that may or may not contribute. So with Target ALS, we sort of created forms that are representative of what we think is most relevant, but the list is not exhaustive, even in what you filled out today. But yeah, not talking about ethnicity because that was an environmental questionnaire. All right, if no other,
I guess I can't help but ask an awfully high-level question, right? So, well, it's been a few decades, right? Gosh, why don't we have more drugs? Why don't we have more drug therapies out there?
Yeah, why don't we have more drugs out there? You mean being tested or just approved or?
Actually, I feel like there's been all kinds of drugs. Maybe the question is more about successful drug therapies.
Yeah, I think it's a couple of different reasons, and so I'm going to give some of my biases too. I think some of the big reasons are that We like to say we know the underlying mechanisms of ALS. The truth is we probably don't, because the way we look at, we're gaining more of them with some of the models that I mentioned, human models that I mentioned. But a lot of what we came up with over the last few decades all came from animal models, which may not be representative of what happens with a human with the disease. Plus, some of them are looking very end stage of the disease where there's so many pathways that are turned on, and they cross all the different neurodegenerative diseases you can think of. And so companies were developing drugs maybe against targets that are in very large populations, and they say, well, that mechanism's in ALS too, let's test it in ALS. That's great. I mean, I'd love to have clinical trials in ALS. The problem is, do we have any real data saying that that mechanism is really important for ALS? Usually the answer is no. The second thing is, yeah, we have all these mechanisms, but the mechanism that's primary for you is different than the mechanism that's primary from you, i.e. precision medicine approaches. That's what I've been trying to push. We need precision medicine approaches, because I can take a blood sample or CSF sample and look at a multitude of biomarkers and start mixing and matching. Oh, this person's more of this flavor, this person's more of this flavor. There's no reason to put this person in a drug that's more favorable here. I already know the answer. It's not going to work. We don't do that. And so when we get the more precision medicine approaches, I think we'll have more successful drug trials in the future.